All right, welcome back. It's time for another deep dive.
Yeah.
And today we're going to be looking at some really fascinating stuff that you sent over.
Okay.
Anti-adhesion therapies and tissue engineering.
Cool.
So it seems like scientists are getting pretty good at controlling how cells stick to each other.
Right.
And, that has some really big implications for medicine.
It's true. Cell adhesion is fundamental to how we build tissues, organs,
Yeah.
pretty much everything in the body.
Yeah.
But you've also highlighted that when it goes wrong,
Right.
it can lead to some pretty major problems like
Yeah.
thrombosis and cancer.
So let's start with the basics here.
Okay.
Cells need to stick together. It seems kind of obvious.
Yeah.
But what's really going on at a molecular level?
So at a molecular level, you can kind of think of it as like cells have these molecules on their surface that act like hooks.
Okay.
They're grabbing onto other cells and the surrounding environment.
Yeah.
And these molecules are incredibly specific. They're almost like a lock and key system.
I see.
And that's what makes cell adhesion such a carefully controlled process.
That makes sense. But then how do things go wrong?
Right.
You mentioned thrombosis,
Yeah.
which is basically blood clots, right?
Exactly. So in thrombosis, certain cells in our blood called platelets
Okay.
get a little too sticky.
Gotcha.
They start clumping together excessively.
Okay.
They form these clots that can block blood flow.
Right.
And it's kind of like a traffic jam in your arteries.
And so that's where these anti-adhesion therapies come in.
Right.
And you highlighted integrin inhibitors specifically.
Yeah.
What are integrin inhibitors?
So integrins are a family of those sticky molecules that we were talking about, and they play a major role in how cells adhere to each other
Okay.
and their surroundings.
Good.
So integrin inhibitors are drugs that block the action of integrins.
Gotcha.
So essentially, they're preventing the cells from sticking together too much.
So they're like breaking up the traffic jam
Ex
in our blood vessels.
Yeah.
You even sent me some information on a specific integrin inhibitor called abciximab.
Right. Abciximab is a really interesting example.
Yeah.
It's used during procedures like angioplasty
Okay.
to prevent blood clots from forming.
Okay.
So basically, it's helping to keep those platelets from getting too cozy and forming a blockage.
That's incredible.
Yeah.
So by understanding how cells stick together,
Yeah,
scientists have actually found a way to prevent dangerous blood clots.
exactly.
What about cancer?
Right.
How does cell adhesion play a role there?
So one of the hallmarks of cancer is uncontrolled cell growth,
Okay.
but it's also about those cancerous cells spreading throughout the body.
Yeah.
And this process is called metastasis.
Right.
And metastasis is largely driven by cell adhesion.
Interesting.
So cancer cells use those sticky molecules
Right.
to detach from the original tumor, attach to other tissues,
Okay.
and then form new tumors.
So it's not just about sticking together.
Right.
It's about them sticking to the wrong things.
Exactly.
And spreading throughout the body.
Yeah.
I see.
Yeah.
So this is where anti-adhesion therapy could potentially play a role in cancer treatment as well.
That's right. So if you can block those specific adhesion molecules
Right.
that the cancer cells are using to metastasize,
Yeah.
then we might be able to slow down or even prevent
Yeah.
the spread of the disease.
That would be huge. So we've talked about how disrupting cell adhesion
Yeah.
can be beneficial in treating thrombosis and potentially cancer.
Right.
But you've also got some fascinating research on how scientists are actually using cell adhesion to engineer new tissues.
Right. So this is where the field of tissue engineering comes in.
Okay.
And it's all about creating scaffolds that mimic the natural environment that cells like to stick to.
Okay.
And this encourages them to grow and regenerate tissue
Gotcha.
in a controlled way.
So these scaffolds are kind of like blueprints for building new tissues.
Yes.
And they rely on these same sticky molecules we've been talking about.
Precisely. So these scaffolds are made of biomaterials
Right?
that are designed to interact with cells
Okay.
in a very specific way.
Yeah.
They provide those essential adhesion cues that cells need
Right.
to attach, grow, and form functional tissues.
So by understanding how cells stick together, we can not only break up those bad interactions,
Right.
but also use that knowledge to build new tissues.
Exactly.
That's pretty mind-blowing.
It really is. And this field is advancing rapidly.
Yeah.
Scientists are developing incredibly sophisticated biomaterials
Wow.
that can mimic the complex structures of different tissues,
Okay.
like bone, cartilage, and even organs.
So we're talking about potentially growing entire organs in the lab.
It might have sounded like science fiction a few decades ago,
Yeah.
but it's becoming more and more realistic.
Wow.
The key is to really understand the language of cells.
Right.
how they interact with their environment,
Mm.
and how we can manipulate those interactions.
Yeah. To our advantage.
This is incredible. We've gone from preventing blood clots to potentially growing new organs,
Yeah.
all by understanding this fundamental process of cell adhesion.
It's pretty amazing.
But you mentioned earlier that this is just scratching the surface.
Right.
There's more to cell communication than just sticking together, right?
Absolutely. Cell adhesion is crucial,
Yeah.
but it's just one part of a much larger story.
Right.
Cells also communicate through these intricate signaling pathways.
Okay.
And it turns out those pathways are more complex than we originally thought.
Okay, I'm intrigued. What's going on beneath the surface?
We sent over some research on a particularly fascinating case
Yeah.
involving heart cells
Okay.
or cardiomyocytes.
Yeah.
And it seems that these cells have different types of receptors
Yeah.
on their surface
Makes
that
sense.
respond to different signals.
So different receptors, different signals.
Right.
What's so special about that?
Well, the intriguing part is that stimulating certain receptors on these heart cells
Increases the force of contractions,
Uh-huh.
while stimulating others does not,
Okay.
even though they both activate the same signaling pathway.
So two different receptors.
Yes,
Same pathway, but different outcomes.
exactly.
That doesn't seem to make sense.
And this is where our understanding of cell signaling gets challenged.
Okay.
You see, the classical view of cell signaling suggests that signals spread evenly throughout the cell,
Right.
like a public announcement.
Gotcha.
But this research that you provided
Yeah.
suggests that there's more to the story.
So it's not just a simple broadcast.
Right.
What else is going on?
Well, it seems that cells might have these tiny localized areas,
Okay.
almost like compartments,
Wow.
where specific signaling molecules are concentrated.
So it's like having different departments within a company.
Yeah, that's a great analogy.
Okay.
These compartments are called microdomains,
Microdomains.
and they allow for much more precise control
Gotcha.
of cellular responses.
So instead of a general announcement.
It's more like targeted messages being sent to specific departments.
Exactly.
So this is where things get even more complex.
Yeah.
Got cell adhesion, signaling pathways,
Right.
and now microdomains.
Yeah.
I'm starting to see why you said we're just scratching the surface.
Absolutely.
Tell me more about these microdomains.
Okay.
How do they work, and why are they important?
One of the key players in creating and maintaining these microdomains is a fascinating group of proteins
Okay.
called akinase anchoring proteins,
Okay.
or AKPs for short.
AKPs. Gotcha.
And think of them like skilled architects
Architect.
carefully arranging the layout of a city.
Wait, architects.
Yeah.
How does that relate to what's going on inside a cell?
It's a great way to visualize it.
Okay.
You see these AKPs act like scaffolding proteins.
Okay.
They're anchoring specific enzymes to precise locations within the cell.
Uh-huh.
And this anchoring helps create those specialized signaling hubs that we were talking about,
Right.
ensuring that signals are delivered and interpreted with incredible precision.
So instead of those enzymes floating around aimlessly,
Why?
they're strategically positioned where they're needed.
Exactly.
That's amazing.
Okay.
But how do they even bind to their targets in such a specific way?
That's where things get even more intricate.
Okay.
AKPs contain this special structure called an amphipathic helix.
Okay, hold on. Amphipathic helix.
Yeah.
Now you're just showing off.
No showing off, I promise.
Break that down for me. What is that and how does it work?
It's actually a pretty simple concept. Imagine a magnet with two poles, one that attracts metal and one that doesn't. An amphipathic helix is kind of like that. One side is attracted to water, while the other side wants to avoid it at all costs.
Okay, that makes sense. So how does that help the AKP do its job?
Well, that water-avoiding side of the helix is what allows it to interact with specific docking domains on target proteins, like protein kinase. It's like finding the perfect parking spot in a crowded lot. The helix snuggles right into that docking domain.
So the amphipathic helix is essentially the AKP's hand, reaching out to grab and position those crucial enzymes.
Exactly. And to make things even more interesting, different types of protein kinase bind to AKPs with different strengths, adding another layer of control to the whole system.
Wow. It's like cells are conducting this incredibly complex symphony of signals, with each player perfectly positioned to create a harmonious outcome. And it's all happening at a microscopic level.
It really is remarkable.
Yeah.
And this brings us back to the puzzle of those heart cells, the cardiomyocytes. The existence of these microdomains helps explain why stimulating different receptors on these cells can lead to different outcomes, even though they activate the same signaling pathway.
So are you saying those receptors are somehow connected to different microdomains within the cell?
That's the current thinking. The beta-1 adrenergic receptors, The ones that boost heart contractions when stimulated with adrenaline are likely associated with a microdomain containing all the necessary signaling components to trigger that specific response. It's like having a dedicated workshop for increasing heart contractions.
And the EP4 receptors, the ones that don't increase contraction force even when activated, are probably linked to a different microdomain that leads to a different outcome, right? It's like a separate workshop with a different function.
Precisely. It's like having two separate workshops within a factory, each equipped to produce a different product, even though they might use some of the same raw materials.
So in this case, the raw material is the GS signaling cascade.
Hmm.
But the final products are different because those signals are processed in different microdomains.
Exactly. And this spatial organization of signaling molecules adds a whole new dimension to our understanding of how cells interpret and respond to their environment.
This is blowing my mind. It's like cells have their own internal GPS system, guiding signals to the right destinations. But I have to ask, how do scientists even study these microdomains? They're so tiny.
It's definitely a challenge. But scientists are developing some incredibly sophisticated techniques. They're using advanced imaging methods to actually visualize these microdomains, and they're also using biochemical assays to study the interactions between signaling molecules within these compartments.
So we're really just starting to understand these microdomains. But the potential implications for medicine are huge, right?
Absolutely. Imagine being able to manipulate cell signaling with such precision that we could redirect signals to promote healing or block harmful processes. The possibilities are truly exciting.
That would be revolutionary. It's like having a microscopic toolkit to fine-tune the inner workings of our cells. This deep dive has definitely given me a whole new appreciation for the complexity of what's going on inside us.
I agree. It's fascinating stuff. And speaking of complexity, you also sent over some interesting research on thyroid hormone receptors, or TRs, and their connection to the VRB oncogene.
Right, I was curious about that. What's
Yeah.
the link between those two?
Well, both TRs and the VRBA oncogene product are part of this larger family of proteins called nuclear hormone receptors. These receptors act as transcription factors, which means they control which genes are turned on or off in a cell.
So they're like master switches, regulating the cell's activity.
Exactly. And what's really cool about TRs is that they can act as both gene silencers and gene activators, depending on whether the thyroid hormone is present.
It's like a two-way switch, controlling gene expression in both directions.
You got it. In the absence of thyroid hormone, TRs bind to DNA and recruit these molecules called corepressors. They essentially act like brakes, preventing the genes from being turned on.
So they're stopping the cell from reading the instructions in those genes.
That's a great way to think about it. But when the thyroid hormone comes along and binds the TR, it triggers a change in the receptor's shape. This change kicks off the corepressors and makes room for coactivators to bind.
And the coactivators are like the gas pedal, pushing the cell to read those genes and produce the proteins they code for.
Exactly. So the thyroid hormone acts as a molecular switch, flipping the TR from a silencer to an activator. It's a remarkably elegant system.
That's incredible.
Yeah.
But how does the VEA oncogene fit into this picture?
Well, the VRBA oncogene product is basically a mutated form of the TR. It's like a switch that's stuck in the off position, permanently bound to corepressors.
So even when the thyroid hormone is present, it can't flip the switch because it's jammed?
Precisely. And this constant suppression of gene expression can have some serious consequences, particularly in the context of cancer.
It's like the VRBA oncogene is throwing a wrench into the cell's machinery, disrupting the delicate balance of gene regulation.
That's a great analogy. And this highlights how important it is to understand the intricacies of these nuclear hormone receptors, both in their normal roles and when things go wrong.
This is getting pretty deep. We've covered cell adhesion, signaling pathways, microdomains, and now these complex gene regulators. It's amazing how much is going on inside our cells that we're completely unaware of.
It really is mind-boggling. And we're only just beginning to unravel all the mysteries. But it's this kind of research that paves the way for new discoveries and breakthroughs in medicine.
Speaking of breakthroughs, I'm curious to hear more about the practical applications of all this knowledge. We touched on anti-adhesion therapies for thrombosis and cancer, and the amazing potential of tissue engineering. But what else is on the horizon?
Oh, there are lots. Well, we've talked about how understanding cell adhesion can lead to new treatments for things like blood clots and cancer, and how it's fueling progress in tissue engineering. But there's so much more.
Yeah, like what What other applications are there? It seems like this knowledge could have really far-reaching implications for medicine.
You're absolutely right. This understanding of cell adhesion and signaling is already leading to new therapeutic strategies for a variety of diseases.
Okay.
For example, researchers are exploring ways to target adhesion molecules in autoimmune diseases,
Okay.
where the immune system mistakenly attacks the body's own tissues.
So by manipulating those sticky molecules,
Yes.
we could potentially reprogram the immune system to stop attacking healthy cells.
Exactly. It's still early days.
Yeah.
But there's a lot of promising research in this area,
Right.
and it goes beyond just autoimmune diseases.
Okay.
We're also seeing progress in using anti-adhesion therapies to treat inflammatory conditions, infectious diseases, and even neurological disorders.
Wow, that's incredible.
Yeah.
It seems like this fundamental understanding of how cells interact is really opening up new avenues for treatment
Yeah.
across a wide range of diseases.
It truly is a game changer.
Yeah.
And it's not just about developing new drugs.
Right.
This knowledge is also helping us design better biomaterials for tissue engineering and regenerative medicine.
Yeah, you mentioned earlier that scientists are already creating scaffolds that mimic the natural environment for cells to grow.
Right.
What's the next frontier in that area?
Well, one of the really exciting areas is 3D bioprinting.
Okay.
So instead of just creating static scaffolds,
Right.
we're now able to print complex three-dimensional structures that more closely resemble the intricate architecture of real tissues.
So it's like taking tissue engineering to a whole new level of precision and complexity.
Exactly. And it's not just about the structure.
Right.
Researchers are also incorporating different cell types and signaling molecules into these bioprinted constructs,
Okay.
creating truly functional tissues that could one day be used to replace damaged organs.
It's like we're on the verge of creating personalized on-demand organs.
Yeah, it's getting there.
That's mind-blowing.
It's definitely a remarkable field with immense potential.
Yeah.
But like any powerful technology, it also comes with ethical considerations
Right.
that we need to carefully navigate.
That's a good point. As we push the boundaries of science, it's important to consider the potential implications and ensure that these advancements are used responsibly.
Absolutely. But with careful consideration and ethical guidance, the future of medicine looks incredibly bright. This deeper understanding of cell adhesion and signaling is giving us the tools to treat diseases in ways we never thought possible.
This has been an incredible deep dive. We've gone from exploring the molecular intricacies of cell adhesion to envisioning a future where we can repair and regenerate tissues with remarkable precision. It's clear that this field is rapidly evolving, and we can expect to see even more groundbreaking discoveries in the years to come.
I completely agree. It's a truly exciting time to be involved in this area of research, and I encourage anyone who's interested to delve deeper into these topics. There's so much to learn and explore.
Well, listeners, I hope you enjoyed this journey into the fascinating world of cell adhesion and its impact on medicine. We've covered a lot of ground today, from the fundamentals of how cells stick together to the cutting-edge applications in tissue engineering and beyond. And as always, we encourage you to stay curious, keep exploring, and keep diving deep into the world of science. Until next time.